Hi guys, let's do section 2. Mendel's laws. So Mendel, the guy that we talked about in the last video, he performed crossbreeding experiments like we just saw where he would cut off the male anthers and pollinate the pea plants himself. He would perform experiments. He used true breeding homozygous plants, which we're going to talk about more in a little bit. So those are plants or organisms whose offspring are going to look exactly the same way that they do. Okay, and he chose plants that differed by only one trait. Pea plants have like 9 traits, I think it is, that are different. So like the height of the plant, the location of the flower, the color of the flower, the shape of the seed, the color of the seed pod. Okay, he chose varieties that were exactly the same except for one of those. And he did something called a monohybrid cross, meaning he was going to breed those plants together, and they were different in only one respect. So mono meaning one, hybrid meaning a combination of the two. So he performed reciprocal crosses. Okay, the parental generation P. Okay, they were bred together to make the first filial generation, which is F1. That F1 generation is then left to self-pollinate or pollinate each other, and you would get the F2 or the second filial offspring. Okay, and this is how he formulated the law of segregation. So here is one of the monohybrid crosses done by Gregor Mendel. This is a true breeding tall plant, meaning if it is left to self-pollinate, every single plant that it makes will be tall, and every single plant that those plants make will be tall. And the same is true for this short one. Okay, it is going to produce all short offspring, and its offspring will produce all short offspring. So these are both true breeding. Okay, that's the P generation, the parental generation. Each one can only pass on one of its alleles, one of its chromosomes. So the tall one is going to pass along a tall allele. The short one's going to pass on a short allele. And they will make a tall plant because this big T, this represents a dominant trait. A dominant trait, when it is present, it will always be represented. Okay? You will always see a dominant trait. So that F1 generation is allowed to self-pollinate, and we get a Punnett square, or a ratio of offspring that is 3 tall offspring to 1 short offspring. And this is probability. It's not exactly, 75% tall and 25. Okay, it's approximate. Okay, so we have 1 true breeding phenotype, one true genotype. Sorry, true breeding genotype. 2 heterozygous, because hetero means different, because they have a big and a little. They have a dominant allele and a recessive allele. And then down here we have a true breeding short, because it has 2 recessive alleles. Recessive alleles only show up when there's no dominant present. So we end up with 3 tall pea plants to 1 short pea plant. So let's talk about the law of segregation. Each individual has a pair of factors, or alleles, for each trait. Those factors, or alleles, segregate or separate during meiosis, the formation of sperm and egg. Each gamete, each of those sex cells, only contains 1 factor from each pair of factors. And fertilization brings those 2 gametes together, and it restores the diploid number, and it now has 2 factors for each trait. All right, so in the results of that monohybrid cross, all of the F1, the filial 1 plants were tall, disproving this blending hypothesis. If the blending hypothesis were true, then the tall plant bred with the short plant would make a medium plant, and that's not what Gregor Mendel saw. In classical genetics and Mendel's cross, Each trait in a pea plant is controlled by two alleles, or alternate forms of a gene. A dominant allele is always represented with a capital letter, and it masks the expression of a recessive allele, which is represented by a lowercase letter. Alleles occur on homologous pairs of chromosomes at a particular gene locus or location. Homozygous individuals have identical alleles on those chromosomes. Heterozygous means that they're different. Here we have a pair of homologous chromosomes. The alleles are located here. Okay, you'll notice that they're in the same spot on the same chromosome. Gene G is here at the top. Gene R is a little bit further down. S is further down from that. And T is all the way down here at the bottom. All right, so replication happens, and we now have two copies. All right, and we know that crossing over is going to occur. We're going to get some exchanging of those genes, and then they're going to separate into 4 haploid cells so that each one has one of these sister chromatids in it. Okay, each gamete has only one, and whenever we put them together with another gamete, we end up with 2. So we have 2 alleles of every trait. Here is a chart showing the different traits that Gregor Mendel studied in the pea plants. Okay, we have the dominant ones here on the left: tall, inflated, round, yellow, axial, purple, and green. Okay, so tall stem length, inflated pod shape, round seed shape, yellow seed color, axial flower position, purple flower, and green pod are all dominant. Okay, if we go over here to the numbers in the F2 generation, you can see that there are way more organisms with those dominant traits than there were recessive. Okay, if we go back to that Punnett square, you'll see that it's roughly 3 to 1. Okay, 2.84 to 1. You round up, that's 3. 2.95 to 1. You round up, that's 3 to 1. Okay, they're all hovering right around 3 to 1. Here we can see our recessive traits are short stem length, constricted pod shape, wrinkled seeds, green seed color, terminal flower, which means it's at the very tippy top, white flower color, and yellow seed pod. Okay, so there's roughly 3 green plants or green pod plants to 1 yellow pod plant. Moving on, genotype. Genotype refers to the 2 alleles an individual has for a specific trait. If those alleles are identical, it is said to be homozygous. If they are different, it is a heterozygous genotype. And phenotype refers to the physical appearance of the individual. Mendel's cross viewed by modern genetics, the dominant and recessive alleles represent DNA sequences that code for proteins. Those proteins are what we can see. That's what gives color, shape, all of those traits. The dominant allele codes for the protein associated with normal gene function within the cell. The recessive allele represents a loss of function. During meiosis I, the homologous chromosomes separate, meaning the 2 alleles separate from each other. The process of meiosis explains Mendel's law of segregation and why only one allele for each trait is in a gamete. Now, Mendel didn't look at just one trait at a time. Sometimes he did what was called a dihybrid cross, meaning he took 2 plants that looked similar in all aspects except for 2. A dihybrid cross uses true breeding plants differing in only 2 traits. Mendel tracked each trait through 2 generations. It started with the true breeding parents differing in the 2 traits. The F1 plants showed both dominant characteristics, and then the F1 plants were allowed to self-pollinate, and he observed the different phenotypes among that filial 2 generation. By doing this, Mendel formulated the law of independent assortment that says that a pair of factors for one trait segregate independently of the factors for other traits. So not all purple flowers are found on tall plants. Okay, you could have a white flower on a tall plant, or you could have a yellow seed pod on a short plant. Okay, they're not directly linked. And the P generation, once again, is the parental generation in the breeding experiment. The F1 generation is the first generation of offspring, and then the F2 is the second generation of offspring. So here we have a dihybrid cross done by Mendel. Don't freak out. We're going to do a lot of practice with these. Okay, we're gonna walk through it together. So we have our two true breeding dihybrid parents. Okay, so we've got dominant here, recessive here. So the dominant passes on two dominant alleles. The recessive passes on two recessive true breeding passes on two recessive alleles. We get two offspring that are heterozygous. Okay, and here we have the ratio of our tall plants with green pods to tall with yellow, and down here we have short with yellow. Okay. Independent assortment segregation during meiosis. So the parent cell has two pairs of homologous chromosomes, and they line up independently of each other, so we can get all of these different variations. All right, this diagram is in your book if you want to take a better look at it. So Mendel's laws, Punnett squares can be used to visualize these traits much more easily. A Punnett square is a table listing all possible genotypes resulting from a cross. All the possible sperm genotypes are lined up on one side, and all the possible egg genotypes are lined up on the other side, and all of the possible zygote genotypes are placed within the squares. Punnett squares allow us to easily calculate the probability of certain genotypes and phenotypes. The Punnett square on the next slide shows a 50% or 1/2 chance of big E and 1/2 chance of little e. All right, the offspring will inherit big E. So there's a 1/4 chance that there'll be big E. There's a 1/4 chance that there'll be big E, little e. There's a 1/2 chance of little e, big E, which is essentially the same thing as this. Okay, and then a 1/4 chance of little e. The sum rule allows us to add the genotypes that produce the identical phenotype to find out the chance of a particular phenotype. So we could actually add these two middle ones together and get 1/2. So it would be 1/4 to 1/2 to 1/4, or 1 to 2 to 1 if we're talking ratios. So here we have the Punnett square that we were just talking about on the previous slide. We're talking about earlobe structure. Here we have an unattached ear and an unattached ear. Okay, and we can actually end up with an attached earlobe given these parents because they're both heterozygous. Test crosses. Individuals with recessive phenotypes always have the homozygous recessive genotype. However, individuals with dominant phenotypes have intermediate genotype. They can be homozygous dominant or heterozygous. A test cross determines the genotype of an individual having a dominant phenotype. So with that dominant trait being present, we don't know if they're homozygous dominant or heterozygous. So here is a one trait test cross. All right, we have a heterozygous parent crossing with a true breeding recessive parent. All right, and we get tall, big T, little t, and our offspring are heterozygous. Two trait test crosses. An individual with both dominant phenotypes is crossed with an individual of both recessive phenotypes. If the individual with the dominant phenotype is heterozygous for both traits, the expected phenotypic ratio is 1 to 1 to 1 to 1. And that's what I've got for you guys. If you have any questions, let me know.